Have you ever thought about the world hiding in plain sight? I'm talking about the massive communities of microbes living on, in, and all around plants. It's so complex, it's basically the plant's second genome. And today, we're talking about how we finally found the key to read it.
So just imagine that for a second. What if we could actually decode this entire second genome? What kind of secrets about how plants grow, how they fight off disease, how they survive, what would we unlock? This isn't just a fun what if anymore. This is the new frontier of botanical research. So, how are we doing it?
Well, for the longest time, our view of this microbial world has been, let's say, a little fuzzy. The old sequencing methods gave us what we call short reads. Now, think of it like trying to read a book by only looking at tiny 150-letter snippets. You kind of get the gist, but you miss the full story, right? It's almost impossible to tell the difference between two very similar characters. But now, modern long reads give us thousands of letters at once. It's like reading entire pages at a time. The difference in clarity is it's a game changer.
So what is this incredible new lens we're using? It's a technology called long Amplicon Sequencing. And believe me, it is completely rewriting the book on the plant microbiome.
Okay, so let's break that down. In simple terms, you start with PCR, which is basically a genetic photocopier. It makes millions of copies of a specific long piece of microbial DNA. Scientists usually go for genes like 16S or ITS because they act like perfect little barcodes for identifying different species. Then, this amazing single-molecule technology reads that whole long barcode in one single pass. This gives us a super high-resolution picture of the entire community.
And the advantages here? Oh, they're massive. Because you're reading such a long piece, you can see right across complex, repetitive parts of the DNA without making mistakes. The accuracy is just phenomenal for spotting tiny little variations that can make a huge difference. And it's a champion at finding big structural changes, stuff that short-read methods would almost definitely miss. Honestly, it's the difference between looking at a blurry, out-of-focus photo and a crystal-clear 4K image.
All right, enough of the theory, right? Let's get into the good stuff. Let's see how this is actually being used out in the field to make some incredible discoveries.
First up, a huge global challenge: drought. Wheat provides something like 20% of the world's calories, but it really suffers when there isn't enough water. So researchers asked a simple question: does the microbiome have anything to do with how some wheat varieties handle drought better than others?
And what they found was, wow, it was striking. The community of microbes living in the soil right around the roots, that's the rhizosphere, was way more diverse for the drought-resistant wheat. And this wasn't just a fluke, it was true no matter where they planted it.
This quote from the study just nails it. It tells us it wasn't just about the soil. The plant itself, its own genetics, were actively recruiting a richer, more powerful microbial support system to help it survive. A more diverse team is literally a stronger team. Okay, next up, let's head for the mountains. Ever wonder how those alpine plants survive just brutal freezing temperatures? Could it be that their microbial buddies are giving them a kind of natural antifreeze? That's exactly what researchers wanted to find out. And the discoveries were just so cool. First, they found the roots were hosting the most diverse bacterial parties. They even identified a core group of bacteria that were present in all these hardy plants. But here's the real kicker. They took these bacteria into the lab, and an incredible 91.5% of them actively helped protect strawberry seedlings from freezing damage. Think about the possibilities there for protecting our crops from frost. And for our final case study, we're talking about ginger. It's a fantastic crop, but it's really vulnerable to a nasty disease called root rot. So the mission here was to figure out the microbial signature of a healthy plant versus a sick one. What does a good soil community even look like? And the answer was just crystal clear. Healthy ginger plants had soils teeming with a high diversity of bacteria. But the diseased plants, they showed a big spike in fungal diversity, which is a classic sign that the bad guys, the pathogenic fungi, are taking over. This tells us that plant health is all about balance. And it could totally lead to things like soil tests that predict disease, or even probiotics for plants to restore that healthy balance. Now, as amazing as this all is, I have to be honest, it's not exactly a walk in the park. Working with this kind of incredibly detailed, long-read data, well, it comes with its own set of challenges that researchers have to figure out. The main hurdles really boil down to three things. First up is data quality. Reading single molecules is powerful for sure, but you can get random errors that need to be carefully cleaned up. Second is just the complexity of the analysis. You get these massive data sets that need some serious computing muscle to process. And finally, of course, there's always the question of cost effectiveness for any research project. So the big question is, how do we get over these hurdles? How do we take this amazing technology from the lab and turn it into real, concrete results that everyone can use?
Well, the answer really lies in having the whole package. It's about combining the right sequencing technology with powerful analysis tools, and crucially, the expertise to know what you're even looking at.
And this is where specialized services can be an absolute game changer. Take a look at this workflow from CD Genomics. It breaks it all down into a super clear 3-step process. You send your sample in. They use cutting-edge PacBio Sequencing to get these incredibly high-fidelity reads. We're talking over 99.9% accuracy, which takes care of that quality issue. And then their expert team handles all the heavy lifting on the complex bioinformatics side.
So what does this actually mean for you, the researcher? It means those big challenges we just talked about are pretty much solved. You get the super high accuracy you need, you can finally see into those tricky complex regions, and you get full gene coverage so you know nothing important was missed. It just accelerates the whole process, letting you focus on the science, not the technical headaches. So we end up right back where we started. We now have the tools to read this second genome with absolutely stunning clarity. From growing crops that can withstand drought to protecting plants from frost, the potential is just immense. So the only real question left is, what secrets in your research are waiting to be unlocked?